Okay, I'm sharing my screen now for the YouTube live session with students' voice. Okay. Now here my camera is sharing means no need of camera share here. Okay, take a minimize. So in the last class, we have seen these kind of things. Okay, we have seen these kind of things. Can you explain about these terms? We have all these things. Just simply explain what these will do. It is a parts of cell. Okay, what these will do in generally.
Um,
What about nucleus? Just give me rough word explanation of nucleus. What happens in nucleus? What its functionality?
it's like the brain. It has like the DNA.
Mhm. Yeah, it stores the DNA. DNA will be available here only, and total genetic information. Genetic information can be uh, stored it here. So entire DNA information, and it is a just like a brain. What we have to do and what is the previous hereditary information. Everything will be stored here only, and here DNA process. How the nucleus bifurcation is also with this nucleus process only. That's cell bifurcation is also happens with the help of nucleus. And here there are some ribosomes. There is some Golgi apparatus, and there is a rough endoplasmic reticulum. There is a smooth endoplasmic reticulum. It is a rough endoplasmic reticulum. There is a smooth. So this outer portion is nothing but smooth endoplasmic reticulum, and this is a Golgi apparatus. We also called the outside, and there are lysosomes, ribosomes, and these are ribosomes, small kind of things. Okay, RNAs, ribosomes, and this is cell membrane. Cell protect. It protects cell. Cell membrane protects the cell, and there are vacuoles where the food storage is processing the mitochondria. It is not the deep cell parts. Okay, it is just a rough cell parts only. And if you get back a little bit previously, so it is a mitochondria. So okay, it's a power storage. Okay, it will control each and everything. And you can see, I think we can see one good image is there here in the previous slides. So yeah, this is a good. It's a proper. One is plant cell, one is animal cell. There is a chloroplast, which is unique in the plant cell, which do the photosynthesis process, and the entire jelly-like thing is called a cytoplasm. And this is a big vacuole in plant cell. There are small vacuoles in the animal cell, mitochondria. Okay, so everything is same. The only unique thing, majorly the unique thing is cytoplasm is available in the cell plants, and in animals are only mitochondria are there. And everything has its own functionality. Everything has its own functionality. And in the there is one more thing. I think in the lysosome we have some functionality table is also here. The nucleus, it contains the genetic information, and ribosomes, it is a protein synthesis, and rough endoplasmic reticulum processes and modifies the proteins. Okay, and these are together, they live together. Rough endoplasmic reticulum, which is covered with small dots. Those are the dots which are ribosomes. So it is a photosynthesis process and processes and modifies the proteins. And smooth endoplasmic reticulum processes the synthesis of lipids. Lipids are fats, you can think. And the Golgi apparatus, it processes and packages the proteins. See, this is a process from ribosomes to rough endoplasmic reticulum to smooth endoplasmic reticulum to Golgi apparatus to send it outside. Lysosomes, then after that lysosomes, and it is packages. Golgi apparatus, it's a processing unit, it's a package unit. Okay, and these lysosomes will digest the cellular waste and materials and foreign matter. Then after that, it will send the outside. If there any unnecessary wastage is there, it will send the outside of the cell. So mitochondria is energy from the organic compounds. See, we take the food, and after the food process, the proteins and nutrients will be made after the synthesis from these above, what is this, ribosomes and rough endoplasmic reticulum. So that energy will be converted into the organic energy, so that your body has to be strong. And the chloroplast, here the photosynthesis process will be happen, and centriole involved in the cell division and formation of cell structures such as flagella and cilia, and they are the tail-like things. It goes to movement of substances across the cell, so how it moves. And vacuole stores the substances also involved in the cell structure in plant cells. It is very great. It is a God gifted for the humanity, because vacuole stores the food in plant cells with a huge amount. So that's why we can see, and this happens in different parts of the plants. Some plants store their food in the root, some plants stores their food in the stem, and some are in leaves, some are in fruits, some are in flowers, and everything is there. Okay, and cell wall, it's a protection. It is a protection purpose. Cell wall. There is no other outer things has to come inside, and it is surrounding by cell membrane, each and everything. Cell wall is crucial. It's important. Okay, and majorly we have to think about the two functionalities: synthesize processing of proteins and lipids, the nucleus, ribosome, rough endoplasm, then it is smooth endoplasm, then Golgi apparatus, then it is the order. It is the order. First of all, nucleus instructs what we have to do. And then after that, ribosome, it's a synthesize. Rough endoplasmic cell will synthesize and modifies the proteins. Then smooth, and if it is not modified by the rough, then it goes to the smooth. Smooth will control the lipids. Then after that, it's the Golgi apparatus, then lysosomes. Okay. Next, energy transformation. Energy transformation means food converted into edible energy. Edible energy means digestive energy, means those your or other organs has to use the energy. So that is the process of mitochondria and chloroplast. Chloroplast is nothing but photosynthesis, and storage and cell structure is nothing but vacuole, plastid, cell wall, cytoskeleton, centriole, cilium, and flagellum. Everything has its own duty, but it is a collective duty of the entire process of this functionality. So then we'll go to the next and next and next. It is the general images which were captured with what is this, microscopes and all. Okay, there are microscopes are also there. There are three to four types of microscopes, because we can't see these kind of bifurcation or dissection in with the naked eyes. So we need some microscopes. Then only we can observe these kind of uh, partition and each and everything. Okay, and this energy transformation. I think you got a good idea how the cell functionality is there. What are the duties of the organisms inside the cell, and this is the process. So now, it is vacuole and cell structures, storage and cell structures. So it collects the food, it collects the energy in one place, in many smaller temporary vacuoles in animal cell and large and permanent vacuole. Animal cell, what happens now? The food will, the food is complete. It is a very small storage and temporary storage. So once the food is collected, then it sends to the ribosomes and everything. It comes to the energy transformation. Then after that, new food will come and new energy will form everything. So that's why when we eat, we get energy. Everything in the process is channeled. Okay. But for this plant cells, na, there is no such a process is there. So the food has to be collected. So that is a big vacuoles. They are the functionality. Next, plastids. Plastids are organelles involved in the synthesis and storage of different chemical compounds. They can contain a double-stranded DNA molecules and are surrounded by a double membrane. Plastids develop from simple organelles called proplastids, animal cells do not contain plastids. So we don't have the animal cells do not have these plastids. It will available only in plant cells. Plant cells. Okay. Chloroplast, which were in. Can you read these 2 points one by one?
Luso Lusoplasts, which are involved in storage.
No, this one.
Oh, sorry. Chloroplasts, which are involved in photosynthesis and are found only in plants and some protists.
Okay. So this is general point. What the chloroplast will do? It takes the sunlight, it takes the water, it takes the carbon dioxide, it forms the carbohydrates. So this is the process of photosynthesis. Okay, and it is available
Yep.
only in the plants. Okay, next. Leucoplast.
Leucoplasts, which are involved in storage. Amyloplasts are a type of leucoplast found in plants.
Okay, next.
They are commonly responsible for synthesizing and storing starch, but can also convert the starch back to sugar when the plant requires energy.
When the plants required energy, so plants also require the energy, but it stores the 100, it uses the 10. That is a way of means it's a general observation I'm saying. So the storage of food is huge, but usage is very less. Okay. See, leucoplasts which are involved in the storage. Amyloplasts are type of leucoplasts found in plants. So means they stores the food. Everything is in what is it, vacuole. Everything is in the vacuole. So they are commonly responsible for synthesizing and storing the starch. Starch is nothing but it is a deep inside of the food particles, but can also convert the starch back to sugar when the plant requires the energy. So sometimes plant requires the additional energy, and the starch becomes to the energy particles. Okay. See, when you see what I can say, when you see carbohydrates. So the carbohydrates cell will be like this, and it will be protected with some other organs also. And there is small thing is there. This is called starch. Okay, it is if it is a one food grain. You can think it is one food grain, and this particle, this core particle, is called your starch. Okay, starch can be you can observe in daily life. Those do you know maida? Maida.
Oh, yeah.
It is the atta is different. Okay? Atta is
Yeah.
different. Maida is different. So you can see the atta without maida because the food companies do they mix all these, they mix together and they make some new product. But maida is not good for human health because it is not good. It's totally different. Maida is not good for the humans. Atta without maida, because maida means it is only the starch. It's the only the starch. So it is different. It is not suggested food for the humans. Okay. So it is internal process only. Internal process. Chloroplast, which contain color pigments and occur in petals and fruits. Chloroplasts have the color pigments. Leaves have the green color. Other fruits have or other leaves have different colors. So chloroplast will maintain that color. Okay, due to this chloroplast only, the leaves are in green, the fruits are in red, orange, yellow, whatever the green color fruits are also there. There are the yellow color fruits. You can see apples, mangoes, and guava in green color, and everything. That is a process of the chloroplast, and with that only we get the food get color. That's a food inside color. Next, cell wall. Cell wall is a rigid structure that surrounds the cell membrane. Okay, so if this is cell, inside is cell membrane. Okay, the outside is cell wall. Cell wall that surrounds the cell membrane of plants, fungal cells, and some prokaryotic cells. It is available in all these things. Okay, I think it is not available in animal cells. In plants, the cell wall is composed mainly of cellulose. Fungal cell walls are made with chitin, some special kind of organism. The cell wall provides the support and prevents the expansion of the cell and allows water dissolve and dissolves the substances to pass freely through it. Lining in the cell walls of woody plants, especially in the xylem, gives them additional strength. Hmm. This is about cell wall. Cell wall is a strong body. See what happens now. In animals, the cell needs to do some more functionality beyond the cell structure, beyond the cell membrane, because it is membrane linked process in the animals. In animals, the cells, the organism of the cells, the movement of cells needs to be more than the plants. In plants, what happens? Wooden is there. Okay, so wooden nothing to do with other organisms and all. That's the strong part of the stem. But for that, how that strengthen because of the cell wall only, because of cell wall. Do you know take? Do you know take? It's a kind of wood. Okay, it's a very strong. It is a very strong. It is a very strong trigger. Okay? So generally, other woods are very weak. Okay, but the take is very strong for the into the your daily home furnitures and all will be made with the take wood. Take wood. Okay, so they are because of their strengthness. It gives more strength to the wood. So that kind of thing is explained here. Cell wall provides the support and prevents the expansion of the cell. So in animal, we need the expansion of the cell. We need the movement of the cell. We need the adjoining and separation of the cells, bifurcation of cells, each and everything we need. But in plants, it is not required. So in plants will contain cell wall, but in animals we don't have cell walls. Okay, and also allows water. Do you know some kind of woods will be processed with sinking in the water, soaken with the water. The wood processing will take a lot of steps in the wood processing, and 1 or 2 days the entire beam will be stored in the water. They will be soaken in the water. Then after that, it will be processed. So every particular kind of wood have their own functionality, and we need its applications and dissolved in substances to pass freely through it and lining in the cell walls of woody plants. Woody plants, take mango tree and lemon trees are there. So their body is very strong, especially in the xylem, gives them additional strength. Due to this additional strength, so it will give the a long stay of the wood part, wood kind of things from the plants. Okay, you can see this is cell wall. This is cell wall, and their yellow colors are cytoplasm and all. Okay, so generally, so everything is very clear in the plant cells and animal cells. And if we go to the next, uh, cytoskeleton. Can you read up this?
The cytoskeleton consists of microtubules
Tube.
of a protein called
Tubulin.
tubulin
It's
and
filamented.
filaments of a protein called actin. The cytoskeleton supports the cell structure, allows the cell to move, and helps transport organelles and vesicles within the cell.
So this is called cytoskeleton. Okay, and it will contain this entire part is cytoskeleton. It will contain microtubulus, and this is a nucleus, and it is a filament. And you can see this is in the cilia and flagella also. Okay, they are the tail-like parts which will be linked with other cells and all. So the cytoskeleton consists of microtubulus of protein called tubulin and filaments of protein called actin. So some kind of protein is called actin. The cytoskeleton supports the cell structures. It is additional support. See, for example, you can think they are the pillars of building. In order to building, they need to stand on pillars. So it is just like things, the cells to be connected with other cells and all. So these will help us. Cell structure allows to cell to move and helps transport of organelles and vesicles within the cell. And centrioles are pair of small and cylindrical centrioles. Small and cylindrical structures compound of microtubulus. They are present in most eukaryotic cells, but many plant cells do not have them. Centrioles are involved in the cell division and formation of the cell structures, such as cilia, flagella. So these are not tube-like things. You can see in the previous image, centrioles. You can see in the previous story, we can go to the previous image. Their centrioles are there, so they help in cell bifurcation. They help in cell bifurcation. These are centrioles. So these tube-like things, they are the tubes. These are centriole. These are centriole, and these are all ribosomes. Again, it will come on centriosome. We call here it is centriosome. Okay, centriole and centriosome. You can now go to the functionality of centriole. Now go to the functionality of centriole. So these are a pair of small cylindrical, just like tubes. Okay, composed of each a single tube is nothing but microtubulus. They are present in most of eukaryotic cells. It's a point, and but many plants do not have them. Centrioles are involved in the cell division. Centrioles are involved in the cell division and formation of cell structures such as cilia and flagella. So what happens in the cell division? So whenever in the last class, I think I have shown that Seneca College material. For example, this is a cell. Okay, particularly consider here nucleus. Nucleus. So in the DNA, it will be condenses these threads, the DNA threads, and then after that it becomes separates the one pair of chromosome, one pair of DNA. Okay, and it goes to the in the next step now it goes to bifurcates, it goes to parts, two parts. Okay, and each will contain one chromosome. Okay, each will contain one chromosome, and they are the haploids, we call it. But in this process, so these centrioles will be help us. Okay, in this process, these centrioles, see, we can explain more, I can explain more, but that is not required here. You have to understand what is the functionality of each and every sub part of the cell is most important. Try to understand and remember the functionality. Okay, whenever you go higher studies, you can understand very clearly. So what the other things, other parts of functionalities of the parts will be helpful. Small and cylindrical structures, and it will be helpful in cell division and formation of cell structures. So this is the formation of new cells. This is the formation of new cells. Cell division. This is the mitosis, meiosis process is there in the DNA division and all. So you can understand there more clearly. Is it clear?
Yeah, it's clear.
So next, these flagellas are the tails, I can say tail-like things or hair-like structures. Okay. Cilia and flagella are hair-like structures on the surface of the cell. So these are the outside. So these kind of hair-like structures are your what is this called cilia and flagella in 2.2.23. So these are the flagella. Okay, so mostly you can see these kind of things in bacteria and fungus. In those two things, you can identify these kind of flagella and cilia. And here, what is the functionality? Can you read at this point?
Cilia and flagella.
Flagella.
Singular,
Flagella.
singular cilium and flagellum are hair-like structures on the surface of cells. They consist of an arrangement of microtubules that are enclosed by an extension of the cell membrane. Both structures are involved in the movement of the cell or things around the cell. Cilia move with an oar-like motion and are usually shorter and more numerous than flagella.
Okay. So it is also same. It is in support of centrioles only. In support of centrioles, small hair-like structures on the surface. They are the outer parts of the cell. Till now, we have seen inner part of the cell. So these will be just like hairs of the cells, and they consist an arrangement of microtubulus, which are from the centrioles, microtubulus, that are enclosed by extension of the cell membrane. It is extension because once the things enter the inside to the cell, after the processing each and everything, proteins, nutrients, photosynthesis, whatever the work done by the cell inside will do. After that, the energy has to go other parts. So that will be via these centrioles and cilia and flagella. So here again, microtubulus and that are enclosed by the extension of the cell membrane. Okay, it is not cell wall. Cell wall is different. It is a strong in the plant cell, but it is linked to cell membrane. Both structures are involved in the movement of cell and things around the cell. So cells should not sit together. They are to be connected with others also. So here the thing, and cilia move with an oar-like motion and are usually shorter and more numerous than flagella. Flagellas are longer and less in strength, and cilia are shorter and more numerous. These kind of more numerous hair will like structures will form. So that is called cilia. And you can see here, this is flagella. Okay, for the bacteria. So these are all in eukaryotic cells. You can get it. Okay. So next we can go to drawing scaled diagram of the cells, and also you need to practice. You need to practice how it looks like and all. Okay, it's a rough line image. Cell wall, cell membrane, it's a plant cell, I think. Okay, whenever cell wall comes, it is a plant cell. Plant cell. Okay, it is a plant cell. Cell membrane, it is a vacuole. It is a entire thing is vacuole. It is a food storage. Nutrients and proteins will be stored here. Okay, it's a big vacuole in the plant cell. And how this is a Golgi apparatus. This is a nucleus membrane. It is a nucleus. This is a nucleus holes. Nucleus. Okay, this is a nucleus. And here endoplasmic reticulum, ribosomes. Some small dots are called ribosomes. This is mitochondria, and there is a chloroplast is also there. This is a greenish in color. Vacuole is a big one. Chloroplast is the other jelly-like thing will be the chloroplast. It's a part of revision of the image of the cell only. And so this is about cells and parts and their functionalities. Just to give me 1 second. I'm putting call. Okay, so it is review. I think I revised 2 to 3 times about the functionalities of all parts. So I will go through this, and we have to go to the next one. What is the next chapter? Next section?
Cell membranes.
Cell membranes. Cell membranes are The cell membrane encloses the contents of cell and controls the movement of substance between extracellular fluids. See, this is your cell. This is your cell inside. This is your cell outside. Okay, inside cell, and this is outside cell, and this is the border, and this is called your cell membrane. This is something are going outside, something are coming inside. It is the main gate, main gate entry. Okay, so you can find here all applications here. Can you read up these first two sentences?
The cell membrane encloses the contents of a cell and controls the movement of substances between extracellular fluid outside the cell and intracellular fluid or cytosol inside the cell. The cell membrane therefore helps maintain an environment within the cell that differs from the external environment.
Okay, so it is just like I can say it is export and imports. Okay, exports and imports. Okay, it is you can say what is that body? Uh, customs department. Okay, as well as it is see we need the communication. Sitting a cell alone one place will not work. That will die. Okay, some energy to come inside, some energy to come outside. It is to be active. So maintain the environment within the cell differs from the external environment. Okay, so that is important, as well as the controlling the transport of molecules into and out of the cell, that is of proteins and nutrients. The cell membrane is also involved in the cell recognition and communication with other cells. In this section, you will learn about the composition of characteristics of cell membrane and is all using the fluid mosaic model of the cell membrane. So extracellular fluids. Extracellular fluids. Mostly water organisms. See, watery organism. Our entire body is wherever cell is there, na, it will look like just like a plasma or liquid mode only. So beyond cell, we need some jelly, some water liquids we need in order to move one place to another place. Okay. Otherwise, it will not sustain. It will not maintain the what is that ecosystem. In order to maintain the ecosystem, something are connected to others. The cells exist in watery environment of extracurricular fluids. Okay, extracurricular fluids. This can be large amount of fluid or just a thin surface layer. Okay, the composition of the extracellular fluids is critical to cell stability and function because it provides them with the nutrients they need to survive, each and everything. In order to survive, they need nutrients and proteins. They will do. They will process. They are the entry gates, entry exit of the cell. The unicellular organisms and extracellular fluids is simply the watery external environments in which they live. They live. Unicellular organisms can do a little or to control their environment. See, basically, bacteria is very basic. Okay, then eukaryotic, then after that prokaryotic. This is the hierarchy of the cells. Okay, so the unicellular, the extracellular fluids is simply the water. In unicellular means only single cellular. Okay, simply the watery external environments in which they live. Unicellular organisms can do a little to control their environment. Bacteria and fungus will not control their environment. They grow and they die. That's it. Okay, but in unicellular, the things to be connected and they need living mechanism. It is not only growing and dying or only growing. It is not like that. That's why fungus can develop. It will not process each and everything, but this the processing is there. So living things, communication is there. So everything we need with the help of cell membrane. Okay, however, some unicellular organisms such as yeast can become dormant until environment conditions are favorable. Other cellular organisms can move slowly to place where the condition suits their needs. For example, unicellular algae can move towards the light, and some bacteria can detect chemicals and then move towards the nutrients or away from the toxic substances. Do you know dormant? What is meant by dormant? Actually, it is a French word because I know it's French a little bit. I complete my French certificate course. What is meant by dormant? If you don't use your bank account in India, if you don't use your bank account for 2 continuous years, your account will be dormant.
Like it's asleep,
It's
basically.
a sleep. It's a sleep. It's a sleeping. It's a sleeping mode. Okay. In French, je dormi means I sleep. Je
Yeah.
dormi. Je means I. Dormi means a sleeping mode. Sleeping mode. So it goes to sleeping mode. So whenever the climate environment, whenever the surroundings are comfortable, then they do activity. Yeast is a good example. Do you know yeast? Yeast, whenever surrounding temperatures are little bit comfortable, then only it will activate. Otherwise, it will not activate. Do you know, in order
Yeah.
to make the breads,
Bread.
and
Yeah.
particularly in South India, we eat what is it, idli and all this. You know, there is a fermentation process is there. Okay,
Yeah.
in order to become a fast fermentation, so the yeast will be added to that maida. Then it will
Yeah.
become ferments easily, and it will become double, and that's where the gas will form. Okay, in the breads, those kind of the gaps with the help of yeast only. So these will not activate immediately according to the environmental conditions. They need some temperature. Okay, then only it will be activate. Otherwise, it will not activate. Otherwise, it will not grow. Even the curd fermentation is also similar. Okay. The other unicellular organisms can move slowly. These will do either sleep or work. Yeast will do either sleeping or working mode. But some others are the slow learners or slow movers. So the unicellular algae. So algae is nothing but your green color thing in the water ponds and all. So when it will happen, according to the sunlight, according to the availability of the sunlight, they develop their growth. So that is also important. And they slowly moves. They slowly moves and they slowly develop according to the sunlight availability. Okay. Some bacteria can detect chemicals and then move towards the nutrients or away from toxic substances. Okay. So according to the ecosystem or surroundings, their movement is there in unicellular organisms and all. Now, extracellular fluids in multicellular organisms. The cells multicellular organisms are more protected from the external environment than the cells of unicellular organisms. Multicellular organisms have more control over the environments in which their cells exist, and therefore less affected by the changes in the external environment. Whether they live in water or land, or multicellular organisms have an outer layer that acts as a protective barrier. The outer layer creates an internal environment. Internal environment means its internal is different climate, external is different climate. So there the cell membrane works very hard. Okay, it is just like you are in sitting in room and standing in outside. Outer temperature is different, inner temperature is different. So both are two different environments. So there the cell membrane works very hard. So this is in the multicellular. This is in the multicellular. And in multicellular organism, the environment of the cells is the extracellular fluids that surrounding them. So outside of the cell, inside of the cell, two different environments followed with the help of cell membrane. And these are commonly, you can see, internal environments and external environments totally different with these factors. With these factors. What is this? One is temperature. Inner temperature is different, outer temperature will be different. Oxygen concentration, inner concentration is different, outer concentration, carbon dioxide, inner, and this will be all in multicellular mainly. And the pH, acidity and alkalinity, and osmotic pressure, concentration of salts and iron, nitrogenous waste concentration, glucose concentration. So they are the main. The way cell interact with the extracellular fluids of the internal environment is regulated by the cell membrane. So everything is the role of cell membrane. It will be controlled by cell membrane as such a strong. See, our body has a proper membrane, proper skin system. It's a totally different. We are studying about the cell only. I'm giving a general example. Moreover, here atmospheric pressure is also most important. Atmospheric pressure is also most important. Of course, with the skin is we need most important. Of course, the even in the body also. What is atmospheric pressure? Pressure in the air on the surface of the earth. What is B pressure? What is BP? This is the blood pressure.
Blood pressure.
They both have the natural restrictions of the organisms. So it is atmosphere means it's totally outside air. The blood pressure means inside your body controlling system. And inside the cell, so these are all. So this is I'm giving a general examples, but cell membrane controls these things. Cell membrane controls cell internal temperature. See, for example, outer side is in the outside of your home, the temperatures are very cool. When you go outside, you feel experience and you will get back. But
Yeah.
but it will not. That is because of your security system. Okay. Even cells also sometimes the outer temperature could be different. At that time also cell to be protected according to the external environment. So for that purpose, your cell membrane will have to work a lot. Okay. And all these temperature, oxygen concentration, carbon dioxide, pH is totally different, acidity and which is basis nature, and osmotic pressure, concentration of salts and iron, and nitrogenous waste concentration. The nitrogen is most important for the plants. Nitrogen most important for the plants in order to their growth and all. In the ratio, they should get the nitrogen according to some ratios and all only, then only the plant will sustain, or general nutrients and all will form, otherwise it will not. Do you know uh, the garden, the terrace garden? Okay, in some countries the terrace garden will be there. So here only water treatment for the plants is there. So there the water should follow, the water should maintain a good amount of nitrogen according to the requirement. High nitrogen is also problem, low nitrogen is also problem. So if we change with these kind of chemicals, no, it will give bad product. Okay, or defective fruits and all will form. That's why we need a natural growth and a possible needed nitrogen proteins and all will be required. Okay, next, cell membrane composition. See, this is your cell. This is your membrane. How a membrane restricts, how a membrane functions. See, where it is happening. This is your entire cell. Okay, these are your cell parts. Each and everything is there. Here it will be available. So it is between the cell membrane and cell wall. This is your part. It is inside. It is cell membrane. Okay. So here the functionality will look like this. Okay, here how this cell membrane, so it protects the cell with these kind of functionalities. What is this? It is carbohydrate chain. It's a proper cell membrane. Okay? It's a polar, non-polar. All these are called poles, and this part is non-polar, and downside is also pole. Okay, cholesterol. Can you see cholesterol? Okay, and
Yeah.
cytoskeleton filament means, see, these are the threads. Hair-like things are cytoskeleton, flagella, and cilia. And these are the proteins. Proteins can come inside and proteins can come outside after the processing. Internal membrane surface. Internal membrane surface means inside. And these are called non-polars. And you can go with some other, it is glycoprotein, external membrane surface. This is the outer part, this is the inner part. Okay, so here your entire cell process is there, and while leaving they need to cross these boundaries with full protection. Is it clear how the cell membrane looks like?
Yeah, it's clear.
It needs a lot of things. It needs a lot of things. See here, inside cell there are ribosomes, and that is rough endoplasmic cell. Everything will be processed. After processed from the each and everything, mostly we have seen several functionalities. So the things has to go outside the cell. So this is the entire process, and these are called your what is it? This is cholesterol. If the cholesterol is huge, the fat will develop. Okay, if the fat will develop, the digestion makes problem. If the digestion makes problem, then the energy makes problem. Okay, and it is not easily digested. They are the lipids. They are the lipids. They are not easily digested. Okay, so this is about the cell membrane image and how it looks, very zoom in very zoom in structure. Okay, so these are the poles, non-polar, and they are the polar. Okay, so how the things are coming inside and how the things are coming outside. ATP. What is mean by ATP? I think I don't know. I don't know. Let's see if it have the definition.
Where does it say that?
No, it is not mentioned here. We'll see. We'll see. We'll see. See, we can see the functionality here. Cell membranes have same basic structure in all organisms. The structure serves to separate the interior of the cell from its external environment. Most membranes are also asymmetrical, meaning one layer has different properties from the other. For example, pattern of protein, carbohydrate molecules in the external surface of is different from the patterns in its internal surface. Okay, means inside is different, outside is different. And here it is the transportation, how the things are coming inside, and this entire big is called enzyme, and it is signaling molecule receptor, and it is signal transduction. We can learn more everything here, but we'll try to understand the terminology. It is cell recognition. See, one cell is recognizing the other cell because they need communication. Okay, and intercellular joining. You can understand it is proper genetics. I will show you in the next class how these cells connect with the same cells, because there is a anchor is there, there is connectivity is there. I will share that PDF, that slides from the genetics portion also, how internally it will be connects. Okay, this cell to cell recognition is important, and intercellular joining. If they are okay, so they can join each other. Okay, and attachment to the cytoskeleton and extracellular matrix. So these are the what is this? This is cytoskeleton, attachment to the cytoskeleton and extracellular matrix. So this attachment it is. Okay, we have a lot of functionalities. We have a lot of functionalities with cell membrane and all. We can learn one by one. Even important. I can't say that you can get everything here only, but uh, by learning in advanced level, you can learn more information. Phospholipids. Phospholipids have molecules have a hydrophilic water attracting head and two hydrophobic tails. It goes to a little bit. Wait a second. This is entire everything is same. Just let me go a little bit inside. Fluidity, cell membrane, and everything will continue. Temperature, the outside. We learn each and everything. With the temperature, whatever the outer body, inner body variations are there. Protein processing, carbohydrates. Okay, let me go with the next class. Okay, I will try to a little bit review and explain more detail.
Okay.
So here I would like to stop here. Here only I'll stop. Okay. So right now it is 3:26 p.m. and we'll continue the next class regarding more about the functionality and how the cell membrane works. What is the in portion, out portion recognition, and process of the cell membrane and all. Let me go to the next chapter also. What we have, investigating cells. Okay. 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 There will microscope. It will come. There are types of microscopes. Electron microscope. It goes to deeper and deeper and deeper. Okay. Calculate the percentages and all. Cell theory. Okay. Review. Again, he is going to revision and all. Okay, we'll continue this portion in the next class and we'll complete, I think, in the next class. I'll stop here. Okay.
Yeah, that's okay.
Control X and take a 10 minutes break. It is 3:27 by 3:40. I will come back. Okay.
Okay.
Okay. Here I would like to stop my live session. Let's go how my class is working there. It is about 2:53 minutes. Let's stop and we'll continue. And here I'm stopping my live session. I don't know how much blocks.